Hi, so thank you for having me. Thank you for picking my talk for the symposium. I'm Ian, I'm a PhD candidate at Cornell University, and I'm excited to talk to you today about supervised learning and about selective sweeps. So when there's an adaptive mutation that rises in frequency, we call that a selective sweep. We classify different sweeps into different modes. So they can be what we call hard or soft, meaning that the adaptive mutation can be associated with only one haplotype or genetic background, in the case of a hard sweep, or with multiple ones, in the case of a soft sweep. And we tend to think that a sweep can be soft, that is, that it can be associated with more than one haplotype, for one of two reasons. Either because the mutation was in standing genetic variation before it became adaptive, or because there's recurrent adaptive mutation at the locus. And not only that, but any sweep of any mode can be driven by weaker or stronger positive selection. So applied population geneticists have gotten quite good at these things we call selection scans, where we search a genome looking for candidate sweep loci. So what I'm interested in is what you would do after that. Right? Once you have found your sweep, how much information about the history of that sweep is actually encoded in the signature that it leaves in the genome? So specifically, can the genomic footprint of that sweep tell us how strong the sweep was, and can it tell us what the sweep mode was? So in other words, whether it was a hard sweep or a sweep from standing genetic variation or a sweep from recurrent de novo mutations. So today, what I'll attempt to do is I'm going to try and tackle these questions by showing you how to infer these sweep parameters from the genomic signatures of known sweeps. And our tool for this is going to be supervised learning. So we're going to be training our inference models with simulations of selective sweeps. So to do our sweep simulations, we're going to be using the software SLIM. So SLIM is a forward-in-time simulator. And that means that it explicitly models every locus of every individual in every generation of the population. And that provides the flexibility that we want, because we want to be able to model positive selection under any demography, life history, or genetic architecture. So our approach starts with reading a coalescent burn-in into SLIM. We simulate the selective sweeps and record the genealogical history of that adapting population in something called a tree sequence. And after this positive selection step is done, we can take that recorded tree sequence and add neutral mutations to it. And that produces the patterns of neutral polymorphism that make up what we call the genomic footprint of the sweep. And with this approach, we can get the best combination of accuracy, flexibility, and computational efficiency. So we're going to be working with a large 1 megabase locus. We take that locus that we simulate and we divide it into 21 overlapping subwindows. In each subwindow, we calculate 7 population genetic summary statistics. And these are statistics that we picked because they're good summaries of both the site frequency spectrum and the haplotype structure within that subwindow. And since sweep signatures can vary in size depending on the strength and the mode of the sweep, we're going to repeat this entire process for 21 different subwindow sizes. Now these different sizes, they can capture different resolutions or zoom levels around the sweep locus. And this should allow us to cover the entire range of possible sweep signatures. And this is what I mean. So here I'm showing signatures as small, medium, and large subwindow sizes. I've highlighted the nucleotide heterozygosity pi here in the thick green. So when a sweep happens, you expect to see a valley of heterozygosity around it. And that means a V or a U shape in the green line. And you can see that for a weaker sweep, the signature looks very clear if you focus on just a few kilobases around the sweep. But it disappears if we zoom out to the full 1 megabase. And for the stronger sweeps in our dataset, the signature actually shows up at a different resolution. So you can't see it at all if you zoom in, but it appears as a white valley when we're zoomed out to the largest size. And we incorporate all these different subwindow sizes in our data at once, so we don't need to worry about how to pick the single best one. For today's results, our simulations are based on Drosophila melanogaster. To get our simulation parameters, we took mutation and recombination rates from the literature, and we combined that with parameter estimates from the DGRP2 dataset. The population size for that dataset we estimate to be in the order of 1 million. So to make our simulations more computationally tractable, we rescale the population size by a factor of about 10 while keeping theta and rho constant. So we actually simulate 2NE of 100,000. And to give our models a variety of sweep scenarios to train on, we simulate a wide range of selection strength and softness. So the range of selection coefficients here might look very wide, but if we take into account our rescaled population size and recombination rate, this actually produces sweeps that range in size from a few kilobases to a few megabases wide, and that's the range of signatures that have actually been observed in nature, including in Drosophila itself. And finally, we set aside 20% of our simulations for model validation, which are the results that I'm about to show you. So to recap, each simulation produces a data structure of 21 subwindows times 21 subwindow sizes times 7 summary statistics. Now, many such simulations form the dataset that we're going to use to train our models to infer the sweep parameters. Our models are going to be convolutional neural networks that are good at leveraging the correlations across these 3 dimensions of subwindow, subwindow size, and summary statistic. So today, I'll show you 2 distinct models. The first is a classification CNN that can detect the mode of the sweep. So that means it'll classify a given sweep signature as one of 3 options: either a hard sweep, or a soft sweep from recurrent de novo mutation, or a soft sweep from standing genetic variation. The random performance for a 3-way classification like that would be 33%, and our model achieves a validation accuracy of 75%. That accuracy rises to above 91% if you focus only on the stronger sweeps in the dataset. And our second model is a regression CNN that estimates the sweep strength for any kind of sweep. So when we validate that with our simulations, we see here in this plot of true versus predicted selection coefficient that the model remains pretty accurate for the entire range of selection strengths. And one thing I want to emphasize is that I'm not presenting here a rigid method. I'm presenting a general approach. So for a new application, you would do simulations that leverage what you know about your organism and your loci. And you would train the models to estimate the sweep parameters that you are interested in. Now, those performances that I've just shown, they represent the best case scenario, right? We're validating our models with a data set that's under the same assumptions as the training data set. In reality, we expect that our training data set will have been misspecified somehow, especially in regards to its demographic history. So what happens when we train our models assuming demographic equilibrium, but the real data set has actually undergone a strong bottleneck that we never knew about? So this is what happens. So as you can see, the regression that estimates the selection strength now has a higher error, and it's making more mistakes because sweep trajectories under a bottleneck look like stronger selection. The classification model that infers the mode of the sweep, however, that one loses some performance, but it remains largely robust to the misspecified bottleneck. And what this does is that this indicates to me that the footprints characterizing selection strength are different than the ones that discern the mode of the sweep. And to test our method and to illustrate to you what a real application looks like, we're going to look at 3 sweep loci in Drosophila melanogaster. Now, all 3 of these loci play a part in the evolution of pesticide resistance, and they can serve as known positive controls because their history is well studied in the literature. We know enough about these loci that we know which alleles are the adaptive ones, and from that information, we also know that all 3 of these loci are, in fact, they're all partial sweeps. So the adaptive alleles range in frequency from around 40 to 76%, so none of them are fixed in the population. Now this is a problem, because the models I've shown you so far were all trained on simulations of fixed sweeps. So we get biased results if we try to apply them to partial sweeps like these. So to address the partial sweep issue, we simulate a new training dataset where sweeps haven't reached full fixation, where they aren't widespread in the population. Now that, of course, will introduce some noise and uncertainty, so we expect that the new models will be less powerful than the original ones that we trained just on the fixed sweeps. But you can see here that the loss of power is small. We can still classify the sweep mode with more than 70% accuracy, and our estimates of selection strength remain unbiased. So these new models are aware of the unique footprints of incomplete sweeps. So we can be a lot more confident when we apply them to our control loci. So let's go ahead and do that. Right? So remember that our models are designed to infer sweep parameters after the sweep loci have been identified. So we took the models trained on partial sweeps that I just showed on the last slide, and we applied them only to the immediate 1 megabase region that surrounds the sweep loci. And these are the results. So all three of our positive controls are soft sweeps. So they're all associated with more than one adaptive haplotype. ACE is a sweep from recurrent point mutations, and its mode is correctly predicted as a sweep from recurrent de novo mutation. Chekhov, on the other hand, is a sweep of a transposable element insertion that was already segregating in ancestral African populations of Drosophila. And that mode is also correctly identified, but as a sweep from standing genetic variation. CYP is the odd one out here, because it's a sweep from recurrent nested transposable element insertions, and some alleles also have a duplication. So it's really unclear which of the two types of soft sweep are going to be the best match for that sort of genomic signature. And our model predicts it to be a sweep from standing genetic variation. And in terms of selection strength, the three loci are all predicted to have strong selection coefficients of around 2 to 4. So to interpret this, we need to take into account that we rescale the population size in our simulations, and that also rescales the evolutionary time. So if you do that, it turns out that these numbers here, they imply sweeps that are a few hundreds of kilobases wide and that have reached their frequencies in a couple hundreds of generations in the Drosophila population. And that fits very well with what we know about the evolution of pesticide resistance in Drosophila. So to summarize, the main thing I want to show you today is that the footprints left by selective sweeps in the genome do carry enough signal to infer sweep parameters. A supervised learning approach is a good way to get at that signal, and I've given examples of models that perform well at inferring both the mode of a sweep and a very wide range of selection coefficients. I've illustrated the approach with an application on pesticide-resistant sweeps in Drosophila, and that application has recovered sweep parameters that are known independently from the literature. And finally, I want to leave you with a concluding remark. So recently, there's been an elephant in the room of population genetics, which is how valid in the first place are machine learning approaches that use simulations. Now that debate hinges on whether simulations represent evolution in nature well enough. And that's something we've been debating for decades, and I'm sure we'll keep debating that for many more decades. But what I hope I've been able to showcase today are some practical benefits of the approach. And the first is that we can make inferences in parts of parameter space that break the assumptions of a lot of the theory that we have. So strong selection is one of those things. And that's something we really want to care about, because very strong positive selection is not only behind the evolution of pesticide resistance like you've seen today, but also behind the evolution of drug resistance in pathogens. And the other main advantage of this kind of method is that you can improve the method by improving your simulations. So you can extend it to new scenarios with very little cost, like we did today for partial sweeps. You can also make inferences that are tailored to the history of the locus you're interested in. So for instance, you could customize your mutation or recombination landscape, you could add background selection, you could add nearby genes, other constraints. Really, you can incorporate in the method anything that you can simulate, which is most things. And this will become more and more important as we discover more hard and soft sweeps in nature that don't behave like we expect them to. And that is all I had to say for today. I want to thank you very much for tuning in. I want to thank my advisors, Philip and Andy, as well as my lab mates and everybody who provided interesting discussion and data. And I'm more than happy to talk about any of this further at the Q&A sessions.